Hi everyone, welcome to the next Biology Required Practical, which is the food tests. So this comes up a lot, not only to do with food, it comes up to do with testing starch in leaves. It might come up in other areas as well. So just be aware of, first of all, the reagent, so the chemical we use and the colour that it goes. Okay, so let's have a look, let's get into it. So we have 4 food tests that we need to know, and you also need to know which foods these are in. So the picture should tell you, and you probably know, but let's just double make sure. So starch, bread, potatoes, rice, pasta, things like that. Sugar, okay, so we've got sugar in tea, but we've also got chocolate and donuts and sweets and all those things. We've got protein. Now they've got a nice slab of, reckon it's beef, reckon it's a steak. But it comes up in other things as well, like beans, and otherwise vegans wouldn't survive. So just be aware of that. Fish, eggs, and milk. There's protein in there. And the last one, lipids, which are fats. So we've got butter there, but also cheese. Be careful if you do food tech, if you say dairy, won't get a mark, okay, in biology. So in biology, we've got fats, protein, sugar, starches. There is no dairy group, if you like. Okay, so we need to know the test for all of these. So we're going to test samples. Obviously, we're not going to do it live here. We're going to test samples to see if they've got these things in them. And what we're looking out for are changes in colour. So these are qualitative tests. It's not telling how much protein or how much sugar's in something. It's just saying a yes or a no. Okay. So we can't measure it as a number, but it will tell us whether it's got it in it or not. So this is an example. We'll look at the reagents in a minute, but no protein is blue, some protein is purple, and lots of protein is like a dark purple. Right, so before we figure out the reagents for the test, so the chemicals we use, you need to prepare your food. Now, there's different ways to doing this, and different exam boards have different kind of marking points for this. So everyone needs to break up their food sample. Everyone, oh look, even get a little demo. Everyone needs to put it in a beaker and add a little bit of water. It's quite hard using chemicals on dry bread or just a lump of potato, which is why we grind it up. Now, all exam boards want those two. Edexcel want this one, which is filter. Okay, and then just use the liquid. So everyone, including Edexcel, if you just learn 1 and 2, there's plenty of other marking points. So break up in a pestle and mortar, put in some water and stir. Now, you only have to do one lot of preparation because what you've made there, you can use for all of the tests now. All right, so let's have a look at one at a time. So there's the extra filter bit. You can put it in if you want to, but it's not always a marking point in all of the exam boards. Right, so using iodine test for starch. So iodine is a reagent, okay, so that's our chemical we're using. And we're going to put about 2 centimetres cubed of this sample that we've just made into a test tube. All right. Going to add a couple of drops of iodine solution. Now you can see from the picture there that iodine is actually orangey colour. So when it goes in, if there's starch, it's going to change colour, we hope. So it's normally browny orange, but if it contains starch, it will be a blue-black. Now, in terms of marking points, what they're looking for is the reagent and the colour change. So this is what you need to write on your revision card, or if you're making notes. Test for starch. The reagent is iodine, and it will go from brown, orange, In fact, you don't always need where it starts. You just need to know that it goes blue back. Blue back. Now, you would get 3 marks for that because it's giving you the reagent and the colour that it goes. And that is a positive test. So this one here, positive test. Now, it's really important you know these 4 enzymes as well when we do enzyme practicals, because you have to be able to test whether the enzyme's working or not. Right, so that's our iodine. Nice simple one there. Let's have a look at the next one. So Benedict's test. So Benedict's is just a blue liquid that comes in a little bottle like this, usually with a pipette. And this is used to test sugars. Now, we can't put it with our food and then put it straight in a flame because it will pop and fizz and all sorts. So we have to use a water bath. So a marking point for this one is you can just say water bath. If you want to add 80 degrees, that's fine, but water bath will get you a mark that you've got a water bath. So set up your water bath. Place some of that filter that we made into a test tube. There we go. Add 10 drops of Benedict solution. Okay, now Benedict solution, as I said, is bright blue. You can see it up here. Where's my pen? There it is. Bright blue colour. And we're going to put it, stand it in the water bath for 5 minutes or thereabouts. And then we're going to note down any change in colour. So this one is a little bit longer than the iodine one. So we've got our water bath. We're going to add our reagent, which is, where are we? There we go, Benedict's. Set it in the water bath for 5 minutes. You don't even have to say 5 minutes, put it in the water bath. Now we need to look at the colour changes. So with this test, at GCSE, you don't distinguish between different types of sugars, you just go sugars. So it can change to lots of different colour, but the rule is, if it changes from blue, it's got sugar in it, but you need to know a couple of the colours that it goes as well. Okay, so here we go. These are our options. So if it stays blue, no sugars. Yellow, orange, green or red, we've got sugars. Now it says higher sugar concentration. It's also to do with the type of sugar, but higher sugar concentration. As long as you know that if there's a colour and it is not blue, it's got sugar in it, or they may ask you what would the colour change be. So you need to know maybe 1 or 2 of these. Okay, you've got quite a selection to choose from here. Okay, so let's just go over it again. Add Benedict's, pop in a water bath, colour changes from blue to either green, yellow, orange or red. That's a positive test for sugar. Right, let's have a look at the next one. Yeah, it says reducing sugars. Don't worry about what they are at the moment because you won't get asked that in GCSE. So. Okay, right, the Biuret test for proteins. Not to be muddled with Biuret, which is a piece of equipment, that long glass tube that you use for titrations. That is spelt Biuret. So be careful. Biuret is the piece of glass equipment. Biuret is the chemical that we use here. So again, we're going to use that sample that we made right at the beginning. Pop it into a test tube. Now it says here add 1 centimetre cubed of Biuret solution A and 1 centimetre cubed of Biuret solution B. Actually, you can just say add Biuret solution and that will still get you the mark. This is what they're looking for, this word. Okay, Bio-Ret solution. There we go. Now, extra mark in this one, give it a shake to mix it up. All right, be gentle though. Don't want it going over your shoulder. Okay, so a swirl is usually best. There we go. Giving it a shake. Note down any change in colour. Right, so the colour for this one, it starts off blue. So you can see up here that Bio-Ret solution is blue. So if it stays blue, no protein. If it goes a lovely shade of purple, and it is a nice shade of purple, we've got protein. Okay, so for this one, our marking points are Bio-Ret solution, shake, and it will go purple. Okay, so that's our test for proteins. Then we've just got one more, which is our test for fats or lipids. Okay, so you can read what it says there. For this test, use the food sample that hasn't been filtered, because actually the filter paper can trap the lipids, so there wouldn't be any in there. So we need to just crush it and put water with it for this one, so that we don't lose all our fats in there. So we're going to place it in a test tube. Here we go. We're going to add a few drops of ethanol. Now, ethanol is flammable, and that's one of their favourite questions. Why do you turn the Bunsen off before you do this food test? Because ethanol is flammable, very flammable as well. I've seen it go up, it's not good. So we need to make sure the Bunsens are off before we do this. Right, there is our ethanol. Now, ethanol is just a colourless liquid, and it goes in. But when it goes in, we're going to give it a shake. Is it going to shake it? Yeah, there we go. Shake it. And note down any change in colour. So this isn't as bold as the other ones. However, what will happen is it will form a white, cloudy emulsion. So it will turn white, basically, and look a bit thick. It'll look thicker than water. So ethanol, shake. Where's my pen? Sorry. Oh, there it is. Shake. We will get a white, cloudy emulsion. All right. So that is our test for lipids. So there's our 4 food tests. So if you've got a revision card, you can. This is a good one here. Chop it into 4. And then in each one, just put the food test, the reagent, so the chemical you need, and the colour it goes. And any other words like shake, something like that, or water bath, add those words too. You can get it on one revision card, what we've just done here. All right, so it shouldn't be too traumatic if it comes up. Now, one word of warning. If you get a 6-mark question about this, please double read it, because sometimes they'll say all of the food tests. Sometimes they'll say protein and sugar. Sometimes they'll say starch and protein. So double check it, because you don't want to be writing things that aren't marking points. Right? Make sure you double check what food test they're asking for. They may not ask for all of them in one go, because that's quite a lot. So they might ask for 2 or 1 or 3. Just double check. Right. Little exam question for you. So pause the video and have a read if you want to, and have a go yourselves. Do that now. Okay. So we've got these 2 food samples. We've got the Bio-Ret test here. No, not here, here. That's testing for protein. So if we look here, look, this one is protein, sugar and starch. Protein, sugar Iodine, you know, tests for starch. Okay. Give 3 conclusions about food A. Oh, interesting. Okay. So food A went blue in the Biuret, it went orange in the sugar, and it went orange, or stayed orange, in the iodine. So let's start with this one. If it stayed orange, food A has, or I won't write it there because I've got the answers to come up in a minute. No carbs. Benedict's test, well, Benedict's is blue, so it's definitely got sugar. And the Biuret test for protein, that starts blue, so no protein. I'll put the answers up in a second. Right, I don't know if I can do this one by one. Yes, there we go. So does not contain protein, does not contain sugar, and does not contain starch. Sorry, does contain sugar, does not contain starch. Right, question B. The student then wished to test the food samples for lipids. Describe how she could do this and what a positive test would show. Right, so it's 2 marks. So we could talk about preparing the sample, but actually it's only 2 marks. So we need to get in there the lipid test, which is ethanol, and the white emulsion. Let's have a look. There we go. Mix the food sample with ethanol and distilled water, if you haven't got it in there already. And the positive test is milky, white, or cloudy. Well done if you got those. Fab job. Right, I'm stopping there and I'm going to do a separate video for this one, which is the required practical, enzymes and pH. Take care everyone. See you on the next one.